Everyone. Welcome to the Human Science Podcast. Now, let me introduce what this is all about so you can see if this is worth your time or not. Recently, I noticed that there's many researchers at our university who are doing truly fantastic work. I cannot learn enough about it, so perhaps I thought others may find it useful as well. With that in mind, I decided that why shouldn't I record it and give access to everyone who might find it interesting? My aim is therefore twofold. First of all, to give you useful and applicable knowledge, but also to give researchers a chance to share what they find interesting and give them a personal interactive medium through which they can share their knowledge. So, with that in mind, I'm pleased to introduce my first guest. She's a brilliant PhD student at the University of Manchester, doing her research into the neuroscience of early life stress. I'm thrilled she's here today. Megan Sheppard, welcome. The first thing that I wanted to get into, because you mentioned that and I thought it was really exciting, is recently you went to Mexico for a month to research a little bit about early life stress, and also I believe there was a national conference. Is that right?
Yeah, so basically, one of the things that we do as PhD students every year is we go to either national or international conferences. So last June, we went to the Organization of Human Brain Mapping, which is also known as OHBM, and they have a yearly conference. So last year, that was in Montreal in Canada. So there was a small group from my lab that went together whilst we were there with my supervisor, Dr. Nils Muller, to give him his full title. We met up with Dr. Eduardo Garza Villarreal from it's called UNAM in Mexico, which is like the big Institute of Neurobiology there.
Mm-hmm.
So he has a PhD student who, because they primarily focus on addiction, but he has a PhD student who has been working on animal models of stress as well as alcohol addiction. So they have already used these neuroimaging paradigms and these behavioral tests to kind of like gather this data. So we realized that my research, which is about early life stress in humans, kind of coincided with their interests, but theirs is obviously in animal models. So when we met there, we decided, okay, let's try and do a big collaboration project. The main aim of it is to kind of see if there are effects that are present in animal models could also be present in humans to see if there's like a cross species effect, basically.
Mm-hmm.
So yeah, we organized a trip. So we applied for an internationalization grant from the university. We got that approved last year. And yeah, in January, I spent pretty much the whole month in Querétaro in Mexico, which was, yeah, absolutely incredible.
Yeah, that's fantastic. So I heard that you guys were working on animal models. You were also thinking later on of working on human models. What is it exactly that you're trying to understand, to research?
So the overall kind of research question is specifically at the minute looking at grey matter volume in the brain.
Mm-hmm.
So the idea is that if you've gone through early life stress, you are obviously having these big traumatic experiences before the age of 18. So my research question specifically focuses on noradrenaline as well. So the idea is that if you've gone through these stresses, you could have these grey matter either increases or decreases in different areas of the brain. And these increases or decreases could be related to the expression of noradrenaline, so noradrenaline receptors. Because that would mean that, if there's an increase, there's likely to be an increase in these receptors.
Yes.
And that could explain potential behavioral differences to stress reactivity. So I've been looking at that in human models for the past year. Um, we are whilst I was in Mexico, we were looking in rat models. And in rat models, you can be a lot more specific. So we can determine much more cause and effect rather than specific relationships. So we were looking at specifically in the amygdala, which is obviously like kind of, the fear center of the brain. We were looking to see if there was grey matter changes in the amygdala.
Mm-hmm.
Now the problem is that the amygdala throughout research, it has shown both increases in volume and decreases in volume. So there's never really like an agreed effect of early life stress in the amygdala. Um, my theory going into this was that the amygdala is made up of lots of different nuclei. So it's not this one brain structure that everyone thinks it is. So I had a theory going in that the two key areas of the amygdala, which is the central nucleus and the basolateral nucleus of the amygdala, that they might be having, there might be different effects on these two nuclei based on obviously if they've gone through early life stress. So it could be that, the grey matter is increasing in the central nucleus whilst decreasing in the basolateral, which is why we can't specify these effects.
So there could be like counter effects.
Exactly, which, they're either drowning each other out or canceling each other out or something like that. So we went in with the rat models because we can determine a lot more specificity in the rat models because
Yeah.
the amygdala is a much larger part of the brain in a rat than it is in a human. So in humans, it's really, really deep in the brain, which makes it a lot more difficult to analyze structurally. But also, it's really, really difficult to then separate the nuclei because how small it is comparative to the rest of the
Oh, I
brain.
see.
So we could use rat models because the amygdala is much larger and we can kind of have a little bit more specificity of effect. So whilst we were in Mexico, we ran, I think, about 13 different models on these rat brains. Um, and we found that there are actually different effects in the different nuclei of the amygdala. So we're currently still analyzing this data, but these grey matter changes that I predicted do seem to be appearing, and it's quite a significant relationship as well. So the next stage of this is we're now going to correlate those volumetric differences found in the two different nuclei,
Hmm.
and we are going to correlate them with the expression of not just noradrenaline, but neuromodulators as a whole. So neuromodulators are basically neurotransmitters, but rather than a single neuron acting on another single neuron, they have what we call volume transmission. So they can stimulate multiple different neurons nearby. So I'm looking to see the four key neuromodulators, which are dopamine, serotonin, acetylcholine, and noradrenaline. I'm trying to see if the expression of those receptors are related to these specific volumetric changes in the rats. Um, and whilst we're doing that, we're also trying to run the exact same experiment on humans, which is obviously a lot more difficult, but we've got the base kind of argument and rationale for that now. So that was it's a summary of what we did in Mexico.
Lovely, lovely. So the way that I understood it, generally what you're finding is that the amygdala, you can actually break it down into even smaller pieces, and that way you'll be able to understand maybe um, how the brain is affected by early life stress.
Exactly. So we can get the specific effects of the different parts of the amygdala. And that might be explaining if the fact that these different parts have different effects, that could be explaining why research isn't converging on a singular explanation of the effects of early life stress. It could be that it's having differential effects on different parts of the amygdala, basically.
Hmm.
Um, so yeah, hopefully that's a preprint, hopefully coming soon.
Yes, yes. I'm counting on it. You mentioned that while you were testing your hypotheses on the
Mhm.
rats, you tested 13 different models.
Yes.
What does that look like exactly?
So the kind of key thing that we do before we go in is obviously we design our hypothesis. That's what we teach you all from day one. You go in with a hypothesis. When you are running what we call modeling experiments, which is where you have, say, a predictive model. So if we're saying we've got two variables that might predict our dependent variable, um, we will have multiple different hypotheses. So the reason we had like 13 different models was because we had very specific hypotheses.
Yeah.
So the first thing we looked at was the overall amygdala. Then we looked at the peaks of the basolateral amygdala. Then we looked at the peaks of the central nucleus of the amygdala. And we then kind of, divided that further by looking at sex-specific effects. So we had kind of like a staged modeling approach where we started broad with the general amygdala, and then we tried to kind of parcelate these models more and see how specific of an effect can we identify? How much of this effect can we provide an explanation for? And
Ah,
that's why we ended up with so many different models.
so if I get you now, it as though you were kind of trying to narrow down, hone in on exactly what parts are doing what
Exactly,
in
and
that case.
exactly what could be driving that change. So, like we said, we looked at sex-specific effects as well because the effects were much more prevalent in male rats than female rats. And so it's kind of not just saying there is something here, there is this relationship, but specifying exactly what can we find out about this relationship. So yeah, we ended up running, I think it was, 13 different models, which was a lot to do in 1 month, but we managed to get it done.
Yeah, I can imagine. Um, so I'm wondering what kind of implications this would have if you did find, I suppose, like early life stress promoting
Mhm.
part of the amygdala, and then the converse of that as well. So in humans, for instance, because you are intending to do it with humans as well, uh, how would that work? What kind of implications could this have on the research more broadly?
So there is a lot of research at the minute into like childhood stress or early life
Yeah.
stress. The difficulty is that like early life stress in general is a real heterogeneous experience. So it could be something as you know much smaller scale. So things like going through divorce. Your parents going through a divorce or marital conflict in the home. That's classified as an early life stressor. There are things that obviously are a lot more rare and kind of larger early life stressors, such as um, neglect, abuse, etc. Um, and there's a lot of unknown about how these different experiences can affect the brain.
Mm-hmm.
And so, if we can find out, okay, in general, here's how early life stress could affect parts of our stress system. So our amygdala is one of our key stress responsivity regions, and it's massively connected in the noradrenergic network in the brain. So it's massively related to our noradrenaline network.
Mm-hmm.
And so if we can kind of elicit what changes are going on in those who've experienced early life stress, we can start to relate this to the behavioral changes that we see in early life stress.
Yes.
So there are two different ways that people can react if they've gone through early life stress. So if you've had significant early life stress, you can either become really, really hypersensitive, where you have these massively maladaptive stress responses to stimuli that shouldn't be eliciting a stress response.
Mm-hmm.
Alternatively, you can become hyporeactive to stress, where all of a sudden you're not responding in a stressful manner to a stimuli that should be stressful.
So you're
So,
desensitized, yeah?
exactly, yeah. So, if we can see, all right, we've got these like structural changes in the brain, the next thing we can do to that is see how that affects the network. So in my instance, I'll be looking at what we call the LCNA network, which is the locus coeruleus noradrenergic network. So the key noradrenaline network in the brain. We can see how that network then changes, and then specifically how those network changes could affect behavior. So it just provides us with a bit more explanation as to why people may react differently to stressors after having these like childhood traumatic experiences.
Mm-hmm. Oh, yeah. So if I'm getting you now, it's as though we are trying to first establish, I guess, the incubation zone of stress and understand, okay, how does it work here exactly in the amygdala? And then you're going to extend it to how does it work more broadly in the network. So if I recall correctly, the LCNA network projects throughout the whole brain.
It does, yeah.
So it you would then be able to understand how the whole brain kind of works in that context, and then
Exactly.
you would be able to link that with more behavioral
Yeah.
uh, like responses.
Yeah, definitely. So we've started looking in the whole brain already. So before I went to Mexico, one of the things that I did was look at gray matter volume changes throughout the whole brain in human models. So my question to you is, have you heard of Biobank?
Yes.
So I use Biobank. Biobank is basically a large-scale medical resource which collected a lot of data from over 500,000 participants in the UK. It collects genetic data, it collects cognitive data. There is anatomical scans of the brain and the body. Um, and as a PhD student, you can apply and pay for access. And then you have data of 500,000 people. So I have analyzed, we started with the limbic system, where we looked at the limbic system in general in humans. And we isolated people who have experienced early life stress. And we looked at the volume changes in the limbic system associated. Um, we did that in 25,000 people.
Yeah.
And again, we found key regions associated with the noradrenergic network that show these changes. We then looked on a whole brain level to see if there was volumetric changes throughout the whole brain. And again, as we said, the locus coeruleus noradrenergic network, the LCNA network, projects throughout the whole brain, and it specifically projects a lot to the frontal cortex, which is your like decision making and
Mm,
associated with a lot of cognition.
okay.
Um, so we have seen massive changes in the orbitofrontal cortex, which is obviously a part of your frontal lobe. Um, and it's just showing how these changes could be connected to noradrenaline, but then these changes could be apparent for specific behavioral paradigms. So that's what we're starting to look in now is like, are these changes not only occurring structurally, but are they occurring network wide? And then what are the potential behavioral ramifications of this?
So that's interesting. So for instance, you mentioned that it projects to the prefrontal cortex.
Hmm.
I know it to be the case that that part of the brain focuses a lot on decision making, on how you, I suppose, discipline yourself.
Yeah, yeah.
So in that case, would that have an effect, or would that suggest an effect for, or would your research suggest an effect for the prefrontal cortex, and would it affect it? How would it affect it?
Yeah, so one massive thing that's associated with early life stress is increased risky behaviors in
Mm,
adolescence and in adulthood. So there is heightened risk of substance abuse, heightened risk of engaging in dangerous behavior. So not quite making the balanced decision making that the rest of us might make.
Yeah.
Now, your frontal lobe is the last part of your brain to develop. That doesn't develop until you're 25.
Yeah.
And so it really does make sense if you think about it logically, why that might be the region that could be massively affected by early life stress. Because obviously, it's going to be the last part to mature. And so potentially, as you're going through these stresses during this maturation period, it could be influencing how it's forming.
Hmm.
Um, there's loads of links between ELS and, early life stress and impulsivity. Um, that then links to these dangerous behaviors, and that could provide an explanation as to why people with early life stress engage in these risky behaviors. This kind of link to impulsivity by the changed and altered structure in the frontal lobe. And then that could link to the early mortality that's associated with early life stress. So you are, I think it's 4 times more likely to die before the age of 70
Hmm.
if you've gone through significant early life stress compared to those who haven't. And a large part of that is because of um, the risks that you take and the impulsive life choices you're more likely to make.
Hmm.
So by looking at this kind of changes in brain structure, it just provides explanatory power as to why there is this relationship between early life stress and increased risk of early mortality.
Mm, that's interesting. So, what I'm getting from what you're saying here, and I'm trying to picture this in like a social context, for example. So, let's say you're born into a worse neighborhood where you're more likely to have
Deprivation.
exposure to these early life stressors. Then I've heard arguments that, maybe you can just get out of it, but it seems as though even your neurobiology was kind of working against you because you have more likelihood to get those early life stressors. And as a result, you also have greater likelihood to be impulsive, even though you may not have, some inherent desire to be that way.
Definitely. But it also links to things like educational attainment.
Hmm.
So if you go through early life stress, you're much more likely to have lower educational attainment. And also you're at much higher risk for increased deprivation in the future. So again, we're providing these kind of links and explanations between, you know, your altered brain structure and your altered brain function that's associated with early life stress, and why this might explain how you then have lower academic performance at school because your brain is maturing at a slower rate than your peers, um, and why um, the more significant the early life stress, the more pronounced these effects are. So it's kind of like you say, it's making the links between how it becomes this perpetuating cycle that's really, really difficult to get out of. Um, and even those who, and it happens more often than not, people are born into deprivation. No one wants to stay in deprivation. No one wants to be making these choices.
Yeah.
So it's kind of demystifying and also taking away the stigma slightly of, you know, people who are born this way will stay this way and all that nonsense that people say.
Yeah.
It's providing an explanation for actually why these relationships exist
Hmm.
and kind of takes the onus and blame off people who've experienced these traumas. It's very, very easy, as people, I mean, I'm speaking for myself, obviously, but who haven't had these early life stresses, um, to kind of become a bit more judgmental about these things. But by providing these links and showing actually your neurobiology is affected, if you're going through deprivation, you're going through these early life stresses, your neurobiology is impacted. So there is only so much that you can do to kind of counteract these effects. And so it really does just provide some kind of like biological explanations for the societal problems that occur as a result. And I just think some of that tries to really help break the stigma of, keeping these cycles going, as people like to say.
Yeah, yeah. Oh, certainly. And I mean, the way that I understand it, this could also have effects on how we make policy. So, for instance, if you understand the mechanisms by which early life stress affects someone's likelihood to be impulsive, then if we're looking for new policing strategies, maybe we should, instead of, you know, mm, creating more violent or more oppressive systems to kind of curb that violence, we should maybe try to find ways in which parents and children can actually coexist more peacefully. And maybe that would kind of project into the future more effectively.
We're also doing some behavioral modeling experiments at the minute that really, really relate to this. So I've been working with Dr. Liz McManus recently, and we've collected some behavioral data on about 90 participants, where we're trying to explore the links between factors like resilience and social support, how they could be protective or, buffer the effects of these early life stresses or adverse childhood experiences, as they're also known.
Hmm.
Um, and so it's looking at how we can kind of help promote social support so that people who've gone through these early life stresses have this support system in place to prevent the cycle from continuing.
Mm,
So, like you say, we could do this on a societal level, which is kind of looking at what implementations can we put in to prevent these effects from happening.
yeah.
And one of the things that we're looking into is whether social support, increasing the social support for people who have experienced these things, will kind of buffer and prevent the potential future implications. So yeah, that's an ongoing study as well as to try and see if we can see behaviorally if there's anything that we can kind of implement to stop these effects from happening as well.
That seems like a fantastic way to kind of integrate the neurobiology and the behavior as well.
Yeah.
So, one thing that I was curious about was you mentioned the social support,
Mhm.
trying to test what is it that would help here. What do you mean by social support exactly? How could I picture this?
So when we're talking about social support, what we mean is, do you have either a network of friends, family, colleagues, therapists, whatever it might be, just a network or even an individual who you can rely on and confide in?
Yeah.
Because a lot of the time with early life stressors, what happens is it's kind of a mental burden that you take on yourself. So, either this has happened to you, or this is an experience that you've had, or, whether that be a one off or a lifelong recurrent early life stressor,
Hmm.
that becomes something that a lot of people keep to themselves. And so what we're testing is if you have someone that you know you can rely on and that you can confide in and that you can kind of have to support you throughout your life, does that buffer the negative effects that are associated with early life stress? Is it a case of having this network of people or this social support will allow you to kind of react differently or, kind of buffer these potentially negative effects, um, and just help kind of improved stress responses in the future?
Yeah. 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 So I can certainly relate to that experience. I've recently been to a um, focus group
Hmm.
where we were talking about how can we make students feel more that they belong.
Hmm.
And I noticed that whenever people, because there were a few students who were consistently said, "Oh yeah, I belong here. I feel that I have a sense of belonging generally." And there were a few others who said, "I don't know. It's like it's hard to find anyone who I can, like you said, confide in or actually have any sort of um, positive experience with." And I noticed that very often they mention how they're more stressed and how they're more, um, I suppose, uncomfortable with their situation and feel that they need to get out of this place. So I can certainly relate to that personally. I mean, I have a million examples where I'd been for a bit alone for quite some time. So definitely felt that way as well. So yeah,
But yeah, that links to like even the teaching side of what we do, which
yeah,
is where, because obviously I do half my time teaching, half my time
yeah,
researching. But that's kind of part of the role of what the GTAs are, which is we're this extra kind of support system in a way to the students where we're one step removed from being your lecturers, your kind of like formal teachers.
yeah,
We're kind of a closer relation to the students. So you feel more comfortable if you have a problem coming to us, or if
yeah,
there's something you don't understand, in class, hopefully you'll feel more comfortable coming to someone who feels more like a peer to you. Obviously, I know that we're kind of still one step removed from you guys,
yeah.
but we're a much closer kind of relation to the students than the lecturers are. So we're kind of here to provide you with some extra kind of academic and social support, and that's where our role kind of fits into your experience. So it's really interesting to see the relationships that we build with the students based on like the social support that we can give you guys.
Oh yeah. Yeah, definitely. I felt that to be the case very often. Like, when you don't have an assignment that you don't know how to do, it's useful to have someone who actually understands what they're doing.
Yeah.
And that gives sense of security, I think, a sense of, I'm not just going to drop out or
Or sit
fail
panicking on my
completely.
own.
Exactly.
Yeah,
Yeah, yeah.
definitely.
Sitting and panicking on your own is not a comfortable
No,
position to be in.
especially when you've got the capacity to ask us these questions and kind of get that help.
Yeah.
It's the best thing to do is just come to us and we'll take you from your breakdown.
Yeah, yeah. Yeah. You'll take us for the edge.
We will.
Lovely. Yeah, so that's something that I wanted to ask you about actually is you being a teacher's assistant alongside your PhD. So the first thing, because I find that, I suppose, career path to be interesting as well. The first thing that I wanted to ask was, you know, how did you choose this pathway rather than, let's say, just focusing on the PhD? Because many students do choose that.
So this is a bit of a difficult one really because the contract that we're on here doesn't actually exist at many other universities. I think there's about three or well, it's getting more and more common, but I only think there was about three or four other universities who offer this kind of a contract when I first started. Um, so what we do is we've got a kind of full-time contract basically where we have half of our time associated with PhD, half of our time has to be directly allocated to either teaching or marking.
Hmm.
Um, Now, I didn't actually know that this path existed. This isn't something that's massively advertised, especially outside of this university, because I didn't go to this university for my undergrad or for my master's. Um, so I didn't know this existed, but the way that this happened to me was that I was doing my master's in Sheffield, and one of my lecturers is friends with my supervisor. And so my lecturer sent out an email because there was only, I think, 30 people on my master's course.
Yeah.
So all of us saying there is a PhD opportunity investigating can't remember what the project was at the time. It's not the project I've ended up doing, which is often the case. Investigating something to do with stress with Dr. Niels Muller at the University of Manchester. So I contacted Niels and I was like, I'm interested, etc., etc. And he's the one who informed me that there are kind of a couple of options you can go down. So the important thing with PhD is to know that there are basically three main options. The first is to get funding from a research council. So in terms of what we do, which is obviously neuroscience or psychology, it's the BBSRC, which is to do with biomedical and biosciences, the MRC, which is like the Medical Research Council. So that's more to do with like the clinical implications or like a disease or a disorder. Um, or you can get more external funding, which will be something like a partner in industry.
Yeah.
Um, Second alternative is self funding. So you are paying your tuition fees, you aren't getting paid to do your PhD. That's all entirely self funded. Now we have our little side door that we have got, which is the third option, not as well known, which is where obviously we get paid a salary to teach and we get our tuition paid for.
Hmm.
So obviously Nils told me about this, and I thought, sounds really good. I quite enjoy um, interacting with people. I really, really enjoy psychology, and I do a neuroscience PhD now. So it's a really, really nice way to kind of keep me with my roots of psychology because
Yeah.
that's what I did my undergraduate degree in. So yeah, it was more that he told me about this, and then the interview process was slightly different because we had to interview for the teaching position first, which we then had to get offered that first, and then apply for the PhD.
Oh, so you had a two-step
We
process.
did, yes. So the PhD interview for me was slightly more informal because I had already had kind of like a pre-interview with Nils. So he was already happy to take me on as a student before I started the teaching process. Um, but yeah, and now I've ended up where I am and very happy for it. It's yeah, really, really fun.
Yeah, yeah. Teaching is a very honorable profession to go into. That's for sure. So that's what I was curious about as well, is when you teach,
Hmm.
when you have the chance to spend time with students
Hmm.
and give them maybe advice or something like that, what do you enjoy about that? Like what do you aim to get out of just being in that situation?
That's a really difficult question. It's a really good question, but it's really difficult because when I teach, I just really enjoy getting to know the students.
Hmm.
Um, I find it really interesting working out which kind of way of explaining things makes the most sense to different people. Um, because obviously there are concepts that we can use that are second nature. So, for example, I was teaching a second year lab today, and I said the phrase, "Oh, this behavioral paradigm,"
Yeah.
and the student turned to me and went, "What's a paradigm?" And I
Yeah.
was like, "Oh my gosh, how do I explain the word paradigm? Like that's not a straightforward thing to explain." And it's just it kind of tests me on my knowledge at times where I always say to students the best way to get a good grade is to take a really really complicated idea and boil it down and make it as simple and straightforward as possible because that shows that you really understand it. And that's what teaching is taking something that's really complicated and making it as simple as possible for students to understand.
Hmm.
And so it's really really good testing yourself to be able to do that. B, it's really, really beneficial for me to see how the different ways that I can boil this down to something simple works differently for different students.
Yeah.
Um, and it's just really nice to get to know you all as well.
Yeah. Yeah.
You all provide different perspectives on things that we haven't considered as well. So yeah, it opens up a good few debates.
That's for sure. Yeah. Yeah. So that was interesting to me. So in a way, students have this just tendency to ask questions of you, which you might have not had the chance to be asked in a graduate position because maybe people already have the sense of what a paradigm is, but maybe maybe you shouldn't have such a like a familiarity, sense of familiarity with it because after all, sometimes they can be more complicated than you think they are.
Exactly. Yeah. Yeah.
So it's like they provide you a greater depth of
Yeah.
understanding.
Definitely. And I
Yeah.
think the depth of understanding thing is where it can be quite difficult as a TA because, for example, we don't get to choose which modules we teach on.
Mm.
Now, our boss, Lizzie Lewis, exceptional woman. I should give her a full title, Dr. Elizabeth Lewis. She allocates all our classes for us. So she takes into account what our PhD specialties are in and then allocates what classes we teach.
Hmm.
Now, timetable means that it's not always possible for me to be teaching the brain and behaviors or the neuroscience or the things that I particularly am strong in.
Yeah.
There are times where I have to teach things like developmental or social psychology, which I really enjoy, but they're not my specialty. And so that brings an additional challenge where it's, information that I know really well because obviously I've learned it myself. I've gone through that program, but it's things that I've not visited in a long time.
Yeah.
So it's really good to kind of like challenge yourself to think, okay, I need to make sure I'm fully prepared for this class, fully prepared to teach it, and kind of going over that information that you just wouldn't touch otherwise. So it really does help kind of teaching the other stuff as well to keep your mind and your perspective a bit wider.
Oh yeah. So definitely that's something that I always found to be the case that when I, let's say, read a book that's a bit outside of what I specialize in,
Hmm.
I get a different sense of those previous ideas, which I
Yeah.
may have tried to explore. So that's something that I'm curious about actually. When you work,
Hmm.
do you have a habit of going outside of whatever it is that you specialize in and trying to read or explore ideas outside of your range?
So that is the what I think is one of the problems with the PhD in general is that
Hmm.
you get caught in your bubble. So a PhD is basically a hyper specialization,
Yeah.
where it's not just specialization in neuroscience. I specialize in the stress of neuroscience, but specifically noradrenergic relations to stress. So that is hyper specific. And so more often than not, you stick in this little bubble
Hmm.
and you only really look within your bubble. It's very, very rare you kind of take a step back and look at the wider, like the wider goings on. So there are two ways that we counteract this. The first for me is teaching.
Yeah.
Um, because, like I say, if I'm teaching developmental labs or something like that, I go, oh my God, that's related to what I do. I've never thought about it from that perspective.
Yeah.
So
Yeah.
taking this step back and teaching the things that I don't specialize in gives me that perspective. But the second way that we do that is by going to the conferences that we spoke about originally. So, for example, this year's OHBM is in Seoul in South Korea,
Hmm.
and we are going in June this year. And what that so the whole point of this conference is it's neuroscience in general, but all of the different speakers are from different topics within neuroscience. So, for example, I do neuroscience and genetics because I use some genetics work in my PhD. And one of the women in my lab is also researching, not similar to me, but still a neuroscience PhD. Um, and because I've been looking into genetics, she's now started thinking about how genetics could influence her project.
Ah.
Um, and by having this variety, so I originally was thinking about genetics partly from going to this conference, which was where people were bringing in the information about genetics and how they kind of incorporate it into what we call imaging transcriptomics, which is mixing, MRI scans or any sort of neuroimaging paradigm with these genetic markers. And so by going to things like these conferences, you start taking ideas from other disciplines and applying them to your work. So there's kind of two different ways that you can take into account other perspectives and stop just keeping in your bubble.
Yeah.
But if you don't do the teaching and you don't go to the conferences, often you miss out on these other perspectives that you could incorporate into your work.
Yeah. It sounds like these conferences, for example, are a bit of like a breeding ground for new ideas where everybody just um those ideas rub against one another
Yeah.
and something new pops out.
Yeah, and collaborations as well, because you end up seeing people, like I said, with Eduardo last year. We
Yeah.
met up, realized that our projects were really, really similar, and we were like, why aren't we collaborating?
Yeah.
It's so silly that we're both doing the same thing, but you're in animal models and I'm in human models. We're not going to do a direct comparison. There's no reason for us to not do that. Um, and so by going to these conferences and stuff, you make those connections where all these ideas that rub together all of a sudden create these new projects and what
That's
we
right.
call our new babies, where we can
Yeah, yeah.
really you know, literally nurture them until they become a fully fledged project.
Hmm. So in conferences like that, is it usually leaders of the industry that not industry, I suppose, of the science that are talking mostly, or is it just anyone who has a discovery that they found?
So the way that it works is you have there's three different types of way that you can speak. So you have got um symposia. So that's obviously you have a theme. So there'll be several parallel symposia running at the same time.
Mm-hmm.
So you'll have a theme which could be something like stress.
Mm-hmm.
And you'll have four different speakers which have 20 minutes each to outline their piece of research. Um, you'll then have a 10-minute Q&A at the end where you can ask them any questions or any clarification.
Mm.
Um, and that tends to be either postdoctoral researchers or professors in the field. Um, Alternatively, we have keynote speakers, which are the only talk that's going on at the time. It's in the big main lecture hall, um, and it's someone who has done something particularly impressive, noteworthy, a big finding in that specific field in the year.
Yeah.
So OHBM that we've spoken about is a neuroimaging conference, so it'll be something specific to an MRI finding or a DTI finding or something like that.
Mm-hmm.
Um, the third way is posters. So this tends to be more for the PhD students. So, for example, last year I presented a poster in Montreal. I'll be presenting another one in Seoul, where we summarize our research onto an academic poster, and people come over to you and do Q&As there. And that's where you can look around at all the different posters that are available and visible, and you can find people who are doing similar things to you and start talking about, again, potential collaborations or potential research that might conflict and why it might conflict. And it just provides an extra kind of avenue of inspiration.
Mm-hmm.
But PhD students can still do things like the symposia, but they're a lot more rare. The poster presentations are more kind of what tend to be done. I'm doing a poster this year, but I'm also chairing what we call an oral session, which I was very surprised to be asked to do, but that was very nice.
Mm-hmm.
Eduardo, who I worked with in Mexico, reached out to me and asked if I wanted to co-chair a session with him, which basically means we'll have a room full of people and a panel of experts. So it's the psychiatry oral session that I'll be chairing,
Mm-hmm.
and it'll be a Q&A session with the experts, and I will be help moderating the questions, kind
I
of
see.
summarizing the answers, rephrasing the questions, etc., for clarity. So yeah, there are loads of different things that go on at the conferences. And it just depends which way you want to address it.
Yeah.
But they're amazing.
So I'm sure that having gone to things like that, or and especially doing research into this, you get a sense of what is this, how is this industry doing, what is it gonna do?
Yes.
So, in your viewpoint, what are the, where do you expect the next big discovery or the next big
Oh, that's a good
insight to be?
question. Um, I honestly don't know. I mean, there's a lot of, like I said, there's a lot of imaging transcriptomics going on at the minute. So that's kind of trying to link the larger scale brain changes that we see in things like MRI to the smaller molecular level changes.
Mm-hmm.
Um, and so that's part of what I'm doing, which is linking the small receptor expression changes to the larger volumetric changes in the brain associated with early life stress.
Mm-hmm.
And I think in the next few years, there will be something massive. I think it will be more to do with a methodology that might come out to link together these, what we call micro scale changes with the macroscopic changes. So the smaller
Okay.
scale to the larger scale changes. I think there's going to be something really interesting happening there.
Yeah, yeah. Um, do you think it's going to be in the field of stress in particular, or is it
I think it'll
possible?
be a more of a data analysis change. So
Mm-hmm.
I think it will be kind of applicable to all the different micro and macro changes.
Mm-hmm.
Um, yeah, I think it'll be more of a tool or a data analysis method that's kind of developed and created
Yeah.
that can kind of help facilitate these like models that are running the analysis stage.
Yeah. Yeah. So kind of the navigation between the small and the large and seeing how they interact with one another. Yeah.
Exactly. And then you can apply that to whatever your research might be.
So I'm curious because, of course, technology has changed pretty much every industry that we go into, including the podcast that we're making right now. But I'm curious as to how was neuroscience affected in the time that you've had to explore it a little bit?
Mhm.
How has technology affected it?
We talk about this quite a lot actually in our lab meetings. So obviously one of the big things that's affecting it is AI.
Mm-hmm.
So for context, I only started neuroscience and neuroimaging in my master's, which was in 2021, maybe. It's relatively recently. So I've
Yes.
only been in neuroscience for about 3 years.
Mm-hmm.
Um, but even since then, AI is changing absolutely everything. Some of the data analysis. So, for example, you can use some AI bots to try and help you with code errors, things like that. So, there are ways that you can use AI to make your life easier in terms of writing your models and doing
Yeah.
your data analysis with your coding. But there are also massive problems now, especially in neuroscience. There have been quite a few controversies lately about using AI to help write articles and using AI to help generate images rather than actually kind of, making your figures yourself and things like that. So there's a big thing in terms of the article, publishing journals and things like that. There's been a big change with AI there. But in terms of research methods and data analysis, there's been a massive shift towards more machine learning approaches and using more kind of like developing algorithms to try and predict changes. Um, and I think it's really, really interesting. But I think it's important we keep kind of the core mathematical modeling that we do, as well as our machine learning models, to make sure that we're not just kind of focusing on the new technology whilst neglecting kind of, tried and true methods.
Yeah.
Um, so, yeah.
So yeah, definitely. I've had a bit of experience making machine learning models.
Hmm.
Just recently, I went to a workshop on it, and oh my God, it's such a fascinating area. But one of the things that I noticed was sometimes there are predictions which you can't really explain. You aren't able to understand them fully. So I suppose one of the problems that you're kind of pointing out here, I guess, is maybe let's still have supervision here. We know you can work on it, and it clearly it has some benefits for, simplifying your workflow.
Yeah.
But still, we need intelligence to
100%.
understand why is it that
I think that's
this
the
is
thing is a
happening.
lot of the time with normal modeling, you go in with like an a priori assumption. This is what I think is the case. This is what I think the relationship will be. And
Hmm.
that this is why I'm hypothesizing it. With machine learning, it's a bit more like, you still go in with these assumptions and you still go in with these hypotheses. Um, but yeah, I think sometimes the interpretation and explanation isn't as clear if you don't go in with as robust assumptions. And I
Yeah.
think that, yeah, that could be a potential problem. But I think they are really trying to, I guess regulate is the wrong word, but make sure that we're doing these modeling approaches in the correct way. Um, and it is all working out. I just want to make sure that yeah, we're definitely keeping our boring old fashioned mathematical modeling going.
That's right. That's right. So mathematical modeling, would that be you using a programming language like Python, for example?
Yes. So obviously at undergrad you learn well here you learn Excel and you learn SPSS.
Okay.
Um, so I learned SPSS while I was at university as well. I did Excel at school, but I use exclusively R,
Yes.
MATLAB, um, sometimes Python, but me and Python don't get along.
Okay.
So yeah, we use our programming languages. So all PhD students tend to we tend to program and code our data analysis as opposed to using a software
Mm-hmm.
that kind of does that code for you.
Mm-hmm. Mm-hmm. Yeah, because from my understanding, programming, you can pretty much procedurize the way by which you make hypotheses. So let's say, if this happens, then this happens. And that's pretty much the way that hypotheses work,
Mhm. But you can also do
like
things
this.
like your sensitivity analyses, so as to say, do we have these extreme outliers?
Mm-hmm.
It's no longer based on a judgment call. Sometimes you have to look at your data and say, okay, I think that's extreme. I'm going to remove it. You
Mm-hmm.
can now code these sensitivity analyses for the program to make that decision
Mm.
for you. So it kind of prevents human error as well.
So like a standardized judgment
Yeah,
by a machine.
exactly. Yeah. So it's, if R doesn't think it should be excluded, then I have no justification to exclude this.
Yeah.
And it also stops essentially p hacking, which is where, people are keep testing and testing and testing until they find the effect they want and then only publishing that effect.
Mm-hmm.
It is a lot more rigorous in that way of, okay, you can't just I'm going to remove this data and remove this data until all of a sudden you have that relationship.
Yeah.
You now say, no, okay, R has told me these data points are all within an acceptable range. I will do my analysis on all of this. And if there's no effect, there's no effect. That's still an interesting finding.
Yeah.
So it just prevents, kind of biased reporting of statistics and p hacking of data.
So in this case, it seems like it does make science a bit more precise, a bit more logical.
Rigorous,
Yeah,
definitely. Yeah,
rigorous.
yeah.
Mm. Yeah, so that's one thing that I was wanted to get to as well is the way that we think of science is, this way of understanding new things and kind of digging in and seeing the causes behind it.
Hmm.
But I noticed that oftentimes, especially when it comes to neuroscience and more, I suppose, specialized industries, where there's seemingly different vocabulary for pretty much everything. and sometimes it can be puzzling to understand, oh, what does this term mean? What does this term mean? And how
Hmm.
do they relate?
Hmm.
So I wonder, in your case, because I assume you're a bit of a science communicator, a PhD student, a teacher. So how would you say that neuroscientists like yourself can make it more accessible to people like me or people that don't know anything about psychology or neuroscience?
I think that's a real problem with neuroscience is that we are hyper specialized. And I think sometimes we forget how hyper specialized it is. So things that might seem like basic principles to us, because we're so hyper specialized, we sometimes misassume that it's common knowledge,
Hmm.
which it isn't. And so I really think that, although I can say terms like fight or flight and know that the vast majority of people will know what I mean by fight or flight, but just provide that clarification immediately afterwards. For those who don't, because a lot of my research is about noradrenaline and changes in noradrenaline, but just by me saying changes in noradrenaline, that doesn't mean that most people would relate that then to changes in your fight or flight response.
Hmm.
And I think it's really, really important as neuroscientists that we're really clarifying what we mean. So, like when I said before, we're using imaging transcriptomics, they're buzzwords. They are just these kind of like really impressive terminology that makes it sound really, confusing and what have you.
Scientific.
Exactly. But that's not the point of what we're doing. We're not trying to do this to sound scientific. And sometimes I think people forget that.
Mm.
The whole point of science communication is for you to understand what we're saying. And that's something I try and do in both my teaching and my PhD, which is, like I said before, how can I explain this in the most clear way possible? How can I make sure you really, really understand exactly what I'm saying? Because it's not just for people who aren't neuroscientists or who aren't psychologists. Other people in my lab do very, very different projects to me, and so don't know things like I was so noradrenaline, for example, okay, is what we call a catecholamine. So catecholamine is just a neurotransmitter that also acts as a hormone. Okay,
Hmm.
it can be um, transferred through the electrical impulses between neurons, but it can also be transferred in the blood as a hormone.
Hmm.
I did a presentation to my lab a while ago, and I said, as we know, noradrenaline is a catecholamine. And my friend put her hand up and went, "What's a catecholamine?" Because it's nothing to do. Her research is completely, completely different. And sometimes it's really good to have people to put you kind of back into the right perspective and think, I'm so pigeonholed. I see this as something that I would assume everyone knows.
Yeah.
But even other neuroscientists who, are insanely intelligent and hyper specialized themselves don't know what this term means because it's not in their field.
Yeah.
So I think that really, really boiling it down and explaining exactly what you mean with everything you say, no matter who your audience is, just means that you really can communicate like your scientific discoveries really clearly
Yeah.
and ensure that they actually understand what you're talking about.
Yeah, that's definitely something I found to be the case. In particular, like when you read a book or when you
Hmm.
try to understand a research paper,
Hmm.
sometimes there's these terms which just puzzle you, but if you skip over them, then you no longer understand the rest of the article. And it's just
Exactly.
impossible to do so. So definitely trying to dig into the definitions of these things, like what is the meaning behind this, not just, I get to learn this term for the exam maybe,
Yeah.
but also what does it mean? Why is it that way? I
Hmm.
think that that's something that definitely whenever I have the chance to do, it's really an empowering feeling.
Yeah.
Like you actually understand things. It
Yeah.
really gives a kick in the discovery,
Yeah.
as
It's
some people
it's
say.
just a confidence boost as well for yourself. If
Oh
you're
yeah,
like, I know exactly what I'm talking about, you just feel much better about it.
yeah,
So yeah.
yeah, yeah. You can use the right words in the right context too.
Exactly, not
So
just putting them in there for the sake of putting them in there.
that's right.
Hugo, I say this when I'm marking assignments. I can
Yeah,
tell if you're putting a word in you don't know what you mean.
that's right.
So as long as you're using words and know exactly what they mean, then you're communicating clearly. And I think if you're using words to try and sound a certain kind of way, you're not going to be communicating clearly anymore.
Yeah, yeah. That's I understand that. So one
Hmm.
more
Of
question.
course.
When you become a PhD student, when you start actually getting to the edges of your field,
Hmm.
you finally have the chance to discover something for yourself.
Hmm.
It isn't just, what does the teacher want me to know?
Hmm.
It's what do I want to know? What is it that, what's really pushing me in that direction?
Yep.
So I'm curious to hear, in your case, how does it feel to discover something new, to try and go into a new direction that no one went before?
It's really exciting. So, like you say, your whole academic career. So, from your undergrad, if you do a master's, if not, sometimes people just go straight to a PhD. But throughout your undergrad and your master's, it is very tailored. You are learning this, and this is what you will know.
Yeah.
As soon as you start your own research and you have agency over that research, it starts to become more important and meaningful to you anyway. Because
Yeah.
again, it's like I say, it's like a baby that you nurture. My projects are my little babies where I'm going, this is something that I have created. This is something that I have designed. I've designed the methodology, I have justified it myself, and I know why I want to do it.
Hmm.
And so when you get that result that matches what you originally thought, or even if it doesn't, and you can rule it out as an explanation, it's still such an exciting feeling. So, like I say, when we were in Mexico, I was working with a PhD student called Helio Rasgado Toledo, who is absolutely excellent. He's a fourth year PhD student. I learned so much from him. And when we got our models back and they were all significant at p's less than 0.001,
Hmm.
we were literally jumping up and down. We
Yeah.
were physically rejoicing. And for people outside of science, they're like, "You absolute geeks."
Yeah.
But that why
What
are
is
you so
P?
excited? Yeah. But for us, it's, oh my gosh, the theories that we had about this specific, hypothesis or whatever you want to call it is actually correct. There is actually evidence to suggest that something that I kind of thought about when I was reading papers and thinking, could this be? I have evidence for it. And it's just such a, yeah, rewarding feeling of, oh my gosh, I actually even just validating, I know what I'm talking about. I've
Yeah.
come up with
Yeah.
this idea and it's not a load of tosh. It actually has some precedence to it. And it does, yeah, it does just give you kind of like a self-affirming feeling as well of like, even if you're not making a difference in terms of like a clinical impact, which is a lot of what we talk about in terms of like psychology, it does make you feel like you're making a difference in terms of the research. You're making a dent into something and you're kind of furthering knowledge in some way. And yeah, it's just so self-affirming.
Well, that was lovely. Thank you very much, Meg. Um, I can't wait to do this again if in case you have time. Thank you very much again and have a great rest
You
of the
too.
week.
Thank you so much.